Okay, so ma'am, good morning, ma'am. We are live now, so we can start ahead with our webinar. And I was really excited from the morning to host you on our platform. So, with this, good morning, everyone. I invite Bhakti Sadguru on the stage, on the mic, to give a light introduction of man. Bhakti, please take the mic.
Thank you so much. Good morning, everyone. Today, I will be introducing Dr. Lipi Thukral, a leading figure in computational structural biology and a principal scientist at CSIR Institute of Genomics and Integrative Biology, New Delhi. Dr. Thukral earned her PhD from the University of Heidelberg, Germany, where she worked on molecular dynamics simulations of protein folding under the mentorship of Professor Jeremy Smith. She further honed her expertise during her postdoctoral research with Professor Saima Khalid at the University of Southampton, UK. In 2012, she was awarded the prestigious DST Inspire Faculty Fellowship and joined IGIB as a computational biologist. Since then, her research has focused on protein membrane interactions, autophagy, and structural bioinformatics, often employing high performance computing to decode complex biomolecular mechanisms. Her significant contributions to the field earned her the CSIR Young Scientist Award in Biological Sciences and more recently the India Alliance supported by DBT and Wellcome Trust. Beyond research, Dr. Thukral plays a pivotal role in India's scientific community as an editorial board member for eLife and Journal for Biosciences, a reviewer for several top-tier journals, and an active member of national expert committees. Her lab, CS Biolab, continues to publish cutting-edge work in protein dynamics, autophagy, and infectious disease structural genomics, often collaborating with experimental and industrial partners to bridge the gap between simulations and real-world biology. With this, I request the host to continue the event. Thank you.
Yeah, Bhakti, really, it was a very nice introduction. Yeah. So, so let's go on with the very first question. During your BSc, what incident or moments prompted you to pursue MSc in bioinformatics? And ma'am, can you please mention the challenges when you are pursuing MSc in bioinformatics during the days 2004 to 2006? At that time, the computers were not that much enriched. So, what challenges and how was your journey regarding your MSc, ma'am? Can you please shed some light on that?
So, um, firstly, I would like to thank the entire student community at the SciComm, and this has been an amazing organization. So, firstly, kudos to you all for putting this consistently good stuff online. And I know you guys are also active researchers, and to pull out people from here and there and to share their views so that somewhere someone can get inspired. So firstly, I want to thank you all for organizing this and also inviting me here at this platform. Coming to the first question, yeah, I think it is, in fact, I was trying to remember in 2004 when I started, and compared to now, we are in 2025. So almost 20 years we are talking about. The change in computational world in general has been massive. And as a consequence, computational biology has also experienced paradigm shifts. So I think mostly the biology was just getting introduced with the word bioinformatics. That was mostly the scenario. There were courses that, in fact, I was a part of the first batch of masters in bioinformatics that I did. Mostly, I remember that's where most of the Indian universities as well as international universities were gearing up with the computational biology. People had made small data sets. In fact, NCBI BLAST algorithms were just there to compare the protein sequences. Now, this was going on, and I was like, how is this possible? How can we study biology through various means? So there used to be an evening course by NIT. At that time, there used to be this a very nowadays as well, but NIT used to offer computer courses, and that was its attempt to, in India as well, all over people used to learn C++ courses. So I was enrolled as a biotechnology graduate in 2001, and absolutely didn't like it doing experiments. So then I was trying to get hold of something, the kind of things that I enjoy. And I've been saying this in the past as well, that that's bachelor time is your moment to explore, because you are an undergrad and you can try to be at different institutes, different internships. I mean, now the world has definitely changed, so you can explore digitally, but at the moment I used to go to industries or to institute. So there was an encounter at the institute where some people were exploring biology through some blast sequences they were trying to understand, and that got me started, that people will need more and more algorithms to handle biological data sets. And for that, one needs to have some understanding of what the languages are. So then I went back and did some evening courses at NIT. And I still remember the infrastructure, the computers were there. And at home, we never had computers. There was this computer which was given only for few hours during the class, and we used to just maximum juice it out and write and learn codes. And at that time, I was I would not be, saying anything lightly, because it's not everything is rosy, but it was almost clear to me that programming and learning biology through computers is how I'd like to pursue. And that's when the next step of masters in bioinformatics happened from bachelor's in biotechnology. So I was very clear to the jump, and during that is where my stint with computers started.
Good. So, ma'am, it was really nice to go and know about your journey. That how did you came across bioinformatics? But when the first time you were introduced with blast sequences, this computational biology, ma'am, what was your inside feelings that how it is possible and all? Can you describe that? Means if we are exposed to something for the first time, we tend to fear that will happen or not. So, what was your situation at that time when you were introduced first?
I mean, you're right. At the first time, I was almost like a kid in the candy store sort of a moment, when you just see a lot of things which you have never seen before, and you're just wondering which one to choose. Because to be honest, there were genes, there were genes as with unique sequences at the genetic level, there were proteins, there were protein structures. Protein structure databases, however, started in 70s also, but at that time it was gaining attention, and people were trying to build uniprot like databases to come together. Then started the attempts to compare the sequences. It was a big, massive ideas of that you can do multiple sequence alignment. And you can do multiple sequence alignment, and you can find out what's same in our species. And those are the questions that kind of make your eyes go huge. So as an attempt to do something, I still remember I made a website. I've taken that off the internet now, but there was a website I made as a tutorial. And that was my way of expressing that how to use these systems. So that was during the masters in bioinformatics. That I still remember I given databases. So what all the databases that are available? What can you do with the sequences? So at every point, I kind of made sure there is an actionable output to my passion or my fascination or my excitement. It just doesn't remain with me. There's a next step to it. So, and I think that website was picked up by many people to make sure I got an entry into PhD. Yeah.
It's really nice when we get exposed to such new things. Even today, I feel really excited when I'm exposed to new things as well. So, ma'am, I got to know that you were appointed as a PR in 2016 and you got a senior position in 2019. So you were establishing your lab at that time. Just then again, sudden COVID pandemic came. So, ma'am, what major challenges you occurred while setting up your lab, and how did you mitigate with?
Yeah, I mean, to be very honest, I had already gained the momentum prior to COVID, so it would be unfair to say that I was setting up during COVID. There were many PIs after me who were doing at setting up
Oh,
post 2020. I was already there at IGIB a couple of years, but when pandemic hits, it was like stopping labs. But at IGIB, we made sure that the institute is up and running. The, the students in my lab were all participating in daily lab meetings because we were actively contributing towards the COVID projects. So, on one hand, we definitely faced challenges related to pandemic, being the cities being shut off, and but at the same time, we also were in a big opportunity where we could do something for the country. We had the genomes with us, and then so as you can see, my lab was continuously publishing in the area of SARS-CoV-2. We wanted to ask the questions of protein dynamics, especially if we can understand how spike protein is oriented on the membrane with respect to different domains, and can we say something by merging genomics, which was coming up every few months with a devastating variant. And the questions were being asked that what can we understand from the protein structures. So I think for me, it was again continuously the scientific aspects that were really in my head during the COVID time. So I don't think I ever went into a shut off mode and tried to, so we compensated along with my entire team being online. And that's the advantage of being a computational biologist that, of course, the other labs had various setbacks. Things could not arrive, and they were seriously hit by the pandemic. But on our front, I think we upped our game by connecting regularly, by meeting, asking questions. And my students, I have to say, they were really a very good bunch. So all of them at that time, we used to daily meet. So there was never a moment where we never discussed science. Of course, there were times when we all went through very, very difficult personal time. So obviously, then we all had to kind of close things. Um, but yeah, it was a very big learning experience for me.
Yeah, really, ma'am. So regarding COVID pandemic, I would like to share one story that with one of my friend Mehak, I was involved in one of your small Twitter post that you gave her some proteins, SARS-CoV-2 proteins to design and art. She's very good at art.
Yeah,
So
yeah,
she made it at art, and I used to convert them into videos that you posted on your Twitter. That I remember.
very nice. Mehak was really, I would say, we had a very short interaction, but she was a very cool designer person who
Yes.
would put ideas in a very simple way. Okay. I hope she's doing good.
Yeah, she's in Germany right now.
Good.
Yeah. So, as you mentioned regarding the long-term outcomes of the research lab, so can you please describe your any work which led to some therapeutic discoveries from your lab or any other current projects which are aimed at developing new therapeutics?
Yeah. So at the moment, I would say we have filed a patent that is on um design protein that can help elevate autophagy. And by doing in this whole process, what we've learned is this that it's very exciting time for protein design and modeling. So what you can do is that you can understand how to engineer a protein based on its structure and dynamics. and you can ask a question, can I just direct it towards a desired function? So that's what we did with one of our recent projects. We are now putting it up also as a manuscript. So this has been a very recent attempt in my lab to now go into therapeutics. But actually, it's very normal in science to take this long uh, to reach a stage where the discoveries can now convert into therapeutic innovations. I was very clear that when I joined to do some fundamental discoveries to understand how autophagosome biogenesis happens, how proteins interact with lipids. And once you do, then you can ask interesting questions. So that's what I think the last few years that we've been asking, that can we now cut off the mechanism which would be required in certain disease scenarios? Can we understand that which interactions can not have other impact in pathways, but just very specific? So this is what we have in the lab, and I'm very excited about that work. Hopefully, that will be out as a manuscript as well soon. But the patent, that all those applications we have done.
So, ma'am, yeah, it's really interesting. So, since you're working in computational biology, I would like to ask, ma'am, with the help of computational biology, is it possible to make therapeutics against against neurodegenerative disorders like Creutzfeldt-Jakob disease, which leads to misfold of the prion proteins?
So that's a very specific question for prion, which is not my field. But if I will take it to a very general question,
Yes, ma'am.
if this is possible, I would just say that now there's no dearth of both industry and academic labs that are making tools, that are giving you very robust tools to play with protein data sets. So now what you can do is that you can ask questions which again are based on protein structure, and earlier these models were not that robust. So as you all know, there was an aha moment with AlphaFold that happened back in 2021, led to a series of tools that have followed in at least the structural biology part. At the same time, also other data sets have been coming in with regard to genomics. There are, I think, and hundreds of genomes available in genomics, which are now pointing towards the variant. So there are so many questions that open up in neurodegenerative disorders. For example, how the variants and how these proteins are different. That's some of the areas where we are working on, and that's where computational biology helps interpret patterns, because it's just so complex, it's not linear. So the way how to merge orthogonally different data sets is also very challenging, right? It just doesn't come up to say a linear path. So I think computational biologists all over the world, I would say there's a lot of excitement in industry where people are asking the same question as you said. So it's no more a question. I think there are now teams that are handling this, that are asking drug development can be accelerated with AI, ML, computational, cloud computing, integrated approaches. And I think it's a very exciting time for people to know how to do programming.
So, ma'am, regarding that, I used to said that computational biology is really a very complex subject. So, ma'am, according to you, how would you make these concepts of computational biology accessible to non-scientific audience?
Yeah, I think uh, this is an excellent question, and actually, people underrate this. People think that it will become obviously very clear, because the moment you are a computational biologist, either the other person doesn't understand you or you don't understand the other person. There's always one way street. So it took me a very long time to, to realize that there are complex jargons above simplicity of the biology. So I think it takes a while. It's not very straightforward to really make complex ideas into simple stuff. I still remember there was a time I like to share that gave me a very critical feedback from, and I'm saying this in as many nice words as I can. There was a scientist who gave a very critical feedback on my presentation when I was a master's student. And this is master's in bioinformatics. And I was trying to tell about ideas, and nothing, absolutely nothing was getting clear in the person's mind. And the feedback was that we don't understand what you're saying, right? And this was me as a master's student. And this feedback is so important, was so important to me, that I put in efforts after that to make sure that my presentations, or when I'm communicating, even in like forums where it is important, that there is great deal of information processed already at my end to explain to a biology audience. For instance, it is not clear how simulations can be targeting time scale. So just to make sure that you have your hands moving. But this I just thought, and my body moved. But imagine that the proteins that are working with this would be operating at nanoscale. So can I just give your attention to saying that what you are saying in experimental biology as fast, which is in seconds, is not fast. It's slow for biology. For humans, at the molecular level, at the atomistic level, the atoms are moving at nanosecond time scale. In fact, there's obviously a picosecond movement as well, but all these collective motions at the time scale can be captured computationally, which can show what is not seen in experiment, and hence becomes critical for understanding dynamic processes. So therefore, these hypotheses should be then combined with experiments to explain what is the mechanism. So this is important. This bit is important to explain that it's not just a fancy computational algorithm.
Yeah, I got goosebumps. Only hearing to the such great things, which I am not exposed to. So yeah, it's really very great to have you here. So regarding that, how do you see the future of computational biology evolving in the next decade?
Mm. For the next decade, I think, I mean, there is an irony to this that while I speak of excitement, there's also a very parallel shuffle which has happened. So, students, especially, are facing a lot of challenges in understanding how to deal with the AI buzz. And I think we are still in a transient state where many labs and many students are trying to grasp what has happened in terms of computational predictions in general. We have LLM models now that can read large amount of data sets. So the future is definitely very exciting, but that also means that all of us have to update ourselves with parallelly what has happened in computational world. And at the same time, there is also an updation of you know cell and gene therapy approaches, CRISPR coming in so rapidly. There's obviously a very clear understanding that we all might just get sequenced very soon. So I think it's it's very exciting, but I think it's also very challenging state for computational biology, because there an era of very tall claims. You know what I mean? So everything can be solved right now, or there is an understanding of the previous times where the pipelines are not yet updated. So I feel that this shuffle is going on. So hopefully we'll come midway in computational biology in coming 10 years, they will understand that it's not a very fast science. कि कुछ करके दे दो. It still needs time. It still needs uh, very thorough understanding of biology. Without that, there is no way that in coming few days we get something very fancy like a pill or something, which is now some of the ways in which the claims are being made that everything can be done with AI. So I think that's also a problematic zone because biology is complex. It needs time. So I'm I'm seeing this extreme end. I'm also seeing the other extreme end where the pipeline and frameworks have not been updated to match what has been going on. So I would say that this is a phase which is a paradigm shift phase that has happened. There's no going back. There's no going back. So the tools and technologies have been vastly updated, but those tools and technologies have to be applied with a meaningful way to answer something in biology. So I think it's going to take some time for all the computational biologists to to kind of create outputs that will outlast us.
Yeah, that's really good. And since you talked about artificial intelligence, so we see the era of computational biology before AI, and now if we integrate AI with computational biology, ma'am, what difference would you see in
Huge. I mean, huge. There is just agentic agents that can be applied to each and every set of problems, and they are being done as we speak. They can be agentic agents just on cancer data sets, and they are being developed right now. So, and those agentic agents are out there in the world doing so many fancy things. So imagine that those agents now coming to biology. What can they do is the answer that we're posing. So the answer is very simple. It will have huge implications. But then, it has to cut various barriers as well, which happens in biology because the cues are different. You and I are processing our food differently. We are in different environment. We are coming with our genetic data set, which is different. And then suddenly, you expect that agents will process this information. So that's what I'm coming to again and again, that it will have huge implications, but because biology is complex, it's going to take some amount of time, which was way, way more time, but now has been reduced drastically because it can process the data in a day. So, you cannot claim to have that done in 6 months now. So, the implications, in short summary for you, is that it's going to drastically reduce the time to understand things.
really? So AI would lead to a biology So, ma'am, as you know, since we are working in science communication, so the networking here we play a role. It's very plays a very crucial role. So, ma'am, can you mention one research project from your lab which you think would have been not been possible without any collaborations in your role as a principal investigator?
Yeah, I mean, if you um, check my publications, most of them are with actively with experimentalists, actively with people in other areas of biology, because I like to have fun with it as well, right? Like if I'm saying something, there has to be one more or many areas in which something which I don't know. And for that, I can't be experts and all. So I really like to work with people. Collaborate extensively. So all of them, you're asking me to almost choose a favorite, that what has been my best
Yes, ma'am.
collaborator, but it will be very unfair because all the so as a scientist, my always favorite work is my most recent work. So because usse pehle whatever is done is done. So right now I am working with my collaborator Dr. Nitin from IIT Kanpur,
Oh,
and he's a physicist by training, and we have had so fun meetings. He has built up his own nanometer imaging. So we proposed what kind of what kind of interactions can happen within protein using simulations. And then we said, Nitin, we have to capture this at nanometer time scale. And then he's working on super resolution imaging with Storm, and that gives us an edge to see the proteins that we propose, the LCP proteins, as clusters, or how they're sitting on the membrane in a physiologically relevant environment. So that's where he comes in, and I think that was critical because whatever we proposed, that how the LCP protein was interacting with PI lipids, we were we had a very strong um, analysis pointing out that they were specific towards a lipid. So they went back and they also had antibodies specific to FIE domain, which refers to the PI lipids, and then they kind of imaged that. So for me, that was a very synergistic and very exciting collaboration, completely complementary, completely synergistic. I found that to be very interesting.
So I'm really, it's really great to have collaboration because they can get the ideas which we cannot.
Exactly.
So, ma'am, as a PhD scholar and postdoc, you have done your PhD and postdoc in various foreign universities, postdoc in foreign universities. So, ma'am, what difference do you see or have you observed in the research culture of Germany, UK, and India?
Yeah. So I did my PhD from Heidelberg University, Germany, but I had a very short postdoc stint in UK. So I would mostly say that the research culture in Germany is largely there's obviously a social differences which keep into the world of your science, so how you do things. But as an Indian, I would suggest that the experiences are different. So you immediately see the work ethics which are different. There is more clarity about what is expected out of you, and that has to be seen as somebody you are responsible with, and this is nobody else's responsibility how the project progresses. This was something which came, because here we are very collaborative in nature, and students and PI spend more time with each other. So there is almost, I would say, the PI and students have more interactions, but outside India, the interaction in general socially is less, so that becomes like a function of time. You realize that the project is like owned by you. I think that's very good. So that's something which I have realized. But overall, um, yeah, I think the expectations change.
That's really great. So, ma'am, since you work on interdisciplinary kind of research, there are some students who think that taking all the four sciences would it be really tough because computation in biology involves physics, chemistry, mathematics, and biology itself. So, ma'am, what do you think on this aspect? That is it easy to handle all the four sciences together, or it's really a bit challenging?
I mean, it depends on how you're looking at it. In 2025, I think this question is not valid because um, the classical departments are going away, the classical chemistry, the classical biology, the classical physics. In other words, I agree with you that it is challenging as a student to develop all kind of expertise. But
Yes.
I would just say that take a day each, and if there is, let's say, a spectroscopy which has come up, which is like, in on the principles of physics, try to understand that particular concept. So it's mostly as you go, these concepts will merge. But if you will say that I will, read the entire physics to understand spectroscopy, is when you will find it very challenging. So the way to go about is this, that okay, these are handful of operations, let's say we have to do. Can I get clarity on the principles of this first? Can I understand what is Newtonian equation of motion if I'm doing in these operations? At least that part should be clear. Can I understand statistical ensembles if I'm doing in these operations? So there are some critical principles that underlie a specific techniques, or the way you're going about things, that would need more of your attention. So if you can just handle one thing at a time, and then probably you're working on drugs that you want to understand how their moieties are. So to have that clarity in terms of chemistry, that what is the basic physical chemical properties of a hydrophobic compound, or how polarity comes down to certain specific pi-pi interaction, something like that. So this, I think, grows as a function of time. So I would request students to not be overwhelmed with everything what they don't know. There is a saying which I say in the lab, just get very comfortable with the unknown. As soon as there is unknown, don't panic. This is normal in science. As a PI, I encountered that daily, so it cannot be that a good scientist, any good scientist I know of, would claim I know everything. So how can we expect that from a student who is venturing into an interdisciplinary project? I think it excites you. You find it like, yes, I'm doing something by just putting two concepts together from different fields. Then you don't need to be overwhelmed that I can't do three different other concepts. So slowly, by each day focusing on your problem, uh, you get better.
Yeah, so, since we talked, we're taking the all four sciences, we need to, for good research, we need to have innovative ideas. So, ma'am, how do you motivate young researchers in your lab to explore innovative ideas?
I mean, see, young researchers anyways have more energy, more their mood, anyways beating with a lot of stuff they can do, because they are the people who are actively working. So I believe they anyways are better. But the only way they get more ideas and get much better with their own selves is by reading. Just one answer I have: the more you read, the more clarity you have about what has not been done, about what are the gaps in the field, and maybe then you also develop a certain kind of courage in pursuing that, because as a student largely the problem is with self doubt. So as a student you always think maybe this is not even a question. So most of the time I feel that young researchers have ideas, but they lack confidence in owning those ideas. They think maybe this has been done, maybe this is not a good question, maybe this is something, if my PI will say or vet it, then only it becomes good. But it's the other way around. The young researchers always have, I think, a new perspective. They just need to read more. This is my request, that as we are going into this technology and a lot of distraction, we don't read as much. So, like your consistency in reading every day in the lab will help you uh, get these so-called innovative ideas that you're saying. It's never a magic. It's always good.
Really, ma'am, it's really great. So, as we are moving towards our end of the webinar, so, ma'am, we ask one question to all our speakers, and what is your favorite discovery in science? For example, if I talk about one of the PhD says that discovery of ATP is a favorite discovery. So, ma'am, what is your favorite discovery in science?
Yeah, I mean, recently I would say our efforts into decade-long efforts into studying this protein LCP has led us to understand that what is the mechanism of this protein in terms of autophagy when it sits on the membrane. So largely people just consider that as a marker, that when that activated, it considered as an autophagy. But I think last few papers that we have in the lab and the current understanding is this, that we know precise motifs of the protein which makes it functional. In fact, that attributes new function to the protein. So I'm very excited about role of membrane interaction of LC3 and how that gives diversity to all the homologs. So I think LC3 protein membrane interactions from the lab is the discovery that I'm very excited about.
Yeah, that's really good. So, last question in our question sets. So, ma'am, what advice would you give to someone looking to lead their own lab?
I think those times are extremely challenging. So you are just only used to doing science, but there are other things that come into picture. And largely, I've seen early career PIs, including me, getting very overwhelmed. Lot of things, which is procuring items for the lab, writing grants, making sure that there is a continuous flow in the of the funds flow, and also understanding how much to network, how many talks to give, what are the areas in which to make a impact. But I would say that prioritize the of what is important. So a person who's opening the lab, the first thing he or she has to make sure is that the core impact, which is that what is your niche area, and what is required to make that science happen is your core. Give yourself 80% time to this. Rest, just keep saying no to other things and only do what is mandatory, at least for the first 5 years. Then your projects in science will get better. So if one can prioritize on the science and not get overwhelmed with, because you can be busy the whole day. That's the whole point. You can, it can be 8 p.m. in the night, and you can come out of the lab and nothing comes out in the direction of your progress. So make sure that doesn't happen. Make sure that you talk out things which you want to make it happen. It can be just one paragraph of Friday. And if that didn't happen, it just will showcase that your priorities were not right for the day. So day after day, hopefully you will learn to prioritize. So my only suggestion is, please prioritize on developing your niche scientific area. Everything else will follow.
That's really good. Yeah. So, basically, what I can summarize from our interaction that being focused, being determined is the and we should be hustle towards our project is the main key for our success. I hope I am right. Or if ma'am you want to add something more into it.
No, you're right.
Yeah. So ma'am, we will be moving on to our YouTube questions. So one YouTube questions we have that in the future trends, how effective will be the molecular dynamic simulations without lab experiments, only experimenting from AI computational models to predict the result of interaction between various biomolecules.
I mean, on its own, um, already MD simulation is able to go for microsecond dynamics. So um, I'm hoping that in future it goes to higher time scales, and also AI will allow probably observing rare conformational transitions, because once you're able to understand the entire trajectory, there are ways in which you can, without going into unbiased ensembles, still see the states which are not seen before. That's definitely one area. And the second area would be this, that the future MD simulations will also, as I said, go bigger in terms of cell scale. So right now, why we have not reached so much of that clarity is because we have smaller systems. So there would be expansion of ways in which we can simulate larger cellular entities. So that will be definitely playing a role with AI as well, because you can use GPU, you can use complex analysis, you can now make more systems with AI, which are possible because of prediction platforms. So there are a lot of possibilities which can be done in this space.
So, ma'am, yeah, really nice. And the next question is that you are working as a project assistant at IIT Delhi after your masters. So, how did this 1 year shape your motivation to pursue PhD in molecular dynamics?
Not in molecular dynamics, but in protein folding. So I think this entire 1 year being in a lab, which was so scientifically motivated, and that environment was created by the professor there, Professor Jairam. I still attribute that lab to everything in my success story, whatever I have done till now, because your initial interactions shape who you are. So I think the whole lab was buzzing. Somebody else was doing MD simulations. I was working on an algorithm called ProRegen, and that algorithm was not related to MD simulations, but I was seeing people doing MD simulations. Right? So there was a constant discussion. So the students in the lab would always discuss, and I always had this in back of the mind what it could do. And then when I appeared for interviews uh, of the possibility of protein folding, which I was doing in my master's internship with the integration with MD simulation, it was a very clear yes to choose among the options I had.
So, the last question, the question we have, is it necessary to do molecular mechanics Poisson-Boltzmann surface area studies and after docking and molecular dynamics simulation studies for publishing a good research paper?
I mean, see, the question is very specific. So what happens is this, that the anything you do after docking, which encompasses energy calculations or MD simulations, it gives flexibility to the complex. So definitely it helps, but it's also dependent on how you are doing it. So I'm not giving a easy yes or no, but it definitely helps, because you're saying that this is docking, which is one snapshot, and now I'm making sure that the protein and the ligand are equilibrated to their dynamics. So maybe there has to be more shape, complementarity which gets covered. So definitely there is an advantage of dynamics, but how long it is being done, what kind of energy and efficiency you're talking about, what are your questions, then they are also very important.
So, the last question of this session, since we are all youngsters and all, we are working towards science communication, like, and there are many other communities. So, ma'am, what advice would you like to give to us so that we can make India a better research country and better country with a scientific aptitude?
Yeah, you are very, very right. There was a recent comment made by a Nobel laureate. I think it is Professor Arnold, if I'm not wrong. She said it during the Lindau Nobel Laureate Committee a meeting where many researchers participate, and that elevates in another understanding of how top class science and communication is also going on. So during that meeting, she said that US is bearing the brunt of not doing communication properly, because most of the masses believe that probably so many technologies that have come up with science are either arbitrary or are not required anymore. And this to me is really shaping how people perceive from so many medias, and uh, when the scientists are not doing their job properly. This was her words, that when scientists do not communicate their ideas properly and take it for granted, that we are getting funding for granted, then this is what happens. So I think the government all over the world, rightfully so, are kind of saying that there has to be an involvement of what you're doing for the society, explain them, right? So there is an almost now, I would say, a part of the science job, and as a function of that, all the students' job as well to communicate what are you doing in your PhDs. Why does it matter? What is the impact of this? And how will it shape future therapeutics? Even if it is long term, what ideas do you have? What are you contributing to the community which was not known? So these are the simple things, if done properly, raises the bar within the country, especially country like India, where some of these discoveries, you know, almost all of us have been like the scientific community, a lot of exposure from people coming outside. So the questions that are we are asking should be communicated to the general public with as much ease as possible. And I think I'm really happy with so many initiatives coming together like this, where people, will see it hopefully in these online forums and try to understand that maybe we are in niche area, but this is important in the bigger pipeline of things which contributes to discovery of a medicine. So it's very important, and I appreciate all the initiatives, including this one in that process.
Really, thank you, ma'am. Since this session was keen to know your journey, was your research experience. So I hope we can get another session with you where we can discuss deep computational biology and how we can collaborate more concepts on computational biology. So this session was kept as a means general thoughts and to know your journey, how you became such a great computational biologist. I'm really very thankful to have you here in our session. So I would request the to please give a vote of official vote of thanks, and I would request Vaishnavi to please uh, give the feedback link in our YouTube chat box for our participants. Please.
Thank you so much. It is my privilege to extend a heartfelt vote of thanks on behalf of all of us here. First and foremost, I would like to express my sincere gratitude to our esteemed speaker, Dr. Lipi Thukral, for sharing their valuable insights and expertise with us. It has left us with valuable takeaways and a fresh perspective. I would also like to thank all my teammates for their efforts in putting together this event so seamlessly. From coordinating the schedule to ensuring a smooth flow, your dedication behind the scenes has made this webinar a success. And a special thanks goes to our audience. Your active participation, insightful questions, and enthusiasm have truly enriched the session. It is your engagement that drives the success of events like this. Once again, thank you all for being a part of this wonderful session. Let's continue to think, grow, and share together. Have a great day ahead. Thank you.
Thank you so much, ma'am.
Thank you guys. All the best. Bye.
Yes. Bye bye. Vaishnavi, please post the feedback link on our YouTube chat box. So, guys, I hope you all have enjoyed it. And let's see. And till then, keep thinking, keep sharing, and keep. And do not forget to like, share, and subscribe our channel, the SciComm Science. And please do fill up the feedback form for getting your e-certificates. We'll see you. Bye bye, everyone.